Okay, good morning. Welcome to the second part of the analysis. In this exercise, what we are going to do is we are going to do enrichment analysis for genes that are associated with different type of p53 mutations, including the R273, R175, and also the nonsense mutation. Okay, first I'm going to show you how you can download the PanCan12 gene mutation dataset from the cancer browser. Okay, to download the dataset, well, first we will go to Google and search for the cancer, we call it cancer browser. Okay, so then just select cancer browser and then you go to cancer browser here. Now, under cancer browser, you can get a lot of the data from the TCGA and then you can download it individually. What you need to do is you go to the add data set here. Now then you can scroll down and there are many TCGA data set down here, but the one that we are interested in would be the PanCan12. So we will click on this one here, and now you can see that okay, there are all the PanCan DNA methylation data set, the gene expression data set, copy number, mutation, protein, and so forth. So we will come back to here to download each single data set in the subsequent exercise. But for this one here, we'll be just looking at the gene level mutation. So all you need to do is you just come here, this button here, and then you press the download button. So then you press OK, and then it will download um, automatically, okay, with the gene, what you call it, the gene mutation uh, data. Now, okay, once you have finished downloaded, you will see a lot of this file here. Well, ignore about the Excel file. So those are the ones that have done the analysis. So uh, the original file that you downloaded and then unzip, you will get a bunch of files, something look like this. So what we will be interested looking at would be this genomic matrix file, which is quite huge. Okay, consists of about 500 over megabytes. Now, if you open this using the text, using the notepad, most likely that it won't work. And even if it works, then if you copy this into the Excel, the original Excel file would not be able to handle such a big, huge data. In fact, when you copy into the existing worksheet, the Excel have a limited number of row and also column. Now, instead of doing that, what you can do is basically you can open your Excel, okay, and then you can actually open this file in the Excel format. So you can go to here and then, well, this is your ordinary Excel. Okay, so I'm going to choose that file that I have already downloaded previously. Okay, which will be BMath Science here. Okay, results and the PanCan12 and then the mutation data set. Okay, you select this. Now you have all these files. So I'm going to open this file here. All you need to do is you just double click and then you open it. So they will tell you that, okay, don't match, but don't worry, you just press the yes button. Now, so you get a bunch of data here. Okay, so it is all tab delimited. So you just press next and then it will automatically arrange it. Okay, separate quite nicely. So it's delimited. So you just press finish and then it will now open. So it will take uh, quite some time to open such a huge file. Sometimes can take 2 minutes, basically. So you just have to be patient about it. Okay, once you open up the Excel file, what you will see is that you have a lot of the 1 and 0 here. Okay, the 1 represent as a mutation and then 0 represent as no mutation. Now, this data only tell you the mutation that are involved in non-silencing mutation. So if there's a silencing mutation, it won't pick up here. So now, on the row here, you will have a lot of the uh, case ID, and then on the column here, each of uh, each of these represent a particular genes and so forth. Now, before we do anything, what we are going to do is we are going to classify each of these case based on their p53 mutation status. Okay, once we have downloaded the mutation dataset from the PanCan 12 database or the dataset, the next thing what we are going to do would be to match each of those cases to the p53 mutation. So this is the mutation dataset that we have downloaded from the cancer genome browser. Now, what I'm going to do now is I'm going to match each of these cases to the known p53 status that we have done in exercise 1. Oh, sorry. What I'm going to do is I'm just going to paste value and then transpose. So these are all the individual cases. Okay. So now then I'm going to open up the file, the class or the classification file, so that we have done in exercise 1. Basically here what we have is we have all the case ID with all the p53 mutation classes that we have designated based on the CBio portal database. So to match all the cases, what I'm going to do is I'm going to use the VLOOKUP function again. So I'm going to look at this. Okay, and then go to the table. So I'm going to select the whole table here. All right. Okay. Then I'm going to return 1, 2, 3. Okay, I'm going to return column number 3 and then false. Okay. So now this will be this. Oh, so you should select here. Okay. See, this will straight away return the tumor type. So then we are going to look at the mutation class. So basically you copy this and then you move to here and change part of the parameters. So now which column is it with the mutation? 1, 2, 3, 4. So I want column number 4 here. So here you go. So then what you need to do is then you can just copy this function and then paste it all the way downward. So then you will be able to match all of this. Now, so here we come back to this whole range of data set here. I'm going to create 2 rows on top here. Mm, yes, okay. So I'm going to create one is the tumor type. Okay, tumor type, and then another one is the mutation. All right. So I'm just going to copy the whole range from here to here, and then I'm going to paste transpose. So now each of the cases will match to a different tumor type now, and then I'm going to do the same uh, for the mutation as well. Okay, right, and then transpose. All right, okay. Okay, so once we have matched all the cases, now what we are going to do is we are going to sort based on the tumor type. So basically to do that, you select the whole data range. Okay, and then after that you go to data and then you go to sort. Now, pay attention to this part here. You need to click on the option here. So it will be, so what we want to sort from left to right and not top to bottom. So we will click this button here. Okay, then we are going to sort from based on row 1 here. All right. Okay, so continue on. So it will take a while because the data set is quite big. So okay, it's done now. So now remember in the first exercise we have decided we are just going to look at 6 different tumors in the PanCan 12 that include the breast cancer, colorectal, head and neck, lung squamous cell carcinoma, ovarian, and also the endometrial cancer. So in that case, so basically we do need all the AML data. We are going to delete all the AML data. So okay. All right, here we go. We are going to delete all this column. Okay, BLCA, we are not looking into BLCA as well. So okay, right, we will delete all the column for the BLCA. Now BRCA, so it's included, so we will remain. CO8 colorectal remain. Okay, GBM not included, so we will delete GBM as well. Okay, head and neck would be remain. Okay, we will remove Kirk. Okay, Kirk and lung adenocarcinoma. So we will remove this. So once we have finished removing all the uh, non-relevant tumor types, so it will end up with a file something like this. Now the next thing what I'm going to do is I'm going to do another round of sorting. So and this time will be based on the P53 mutation status, and basically those mutations that are not is not interested in our study, I'm going to exclude that as well. So I'm going to do it again. So now this time will be sort by row number 2. Okay. So it will again take some time. Okay, here you go. So in our study, basically we are not interested in frame swift mutation. We can delete all the frame swift mutation here. Okay, I'm also not interested in in-frame deletion or other missense mutation as well. So we will be removing quite a lot of the data actually. Okay, all the way to missense. Multiple mutation. Nope, I'm not interested in that as well. So basically all this would be delete. Okay. And then NS, nonsense mutation, I'm interested in it, so will remain. And then other types of mutation, no, not necessary. We are going to remove it. R175 remain. Okay. Now R248, well, I think for this study we are not looking into it, so we will remove it. But honestly speaking, I think R248 is another hotspot mutation, which will be quite interesting to analyze in the future. Anyway, for this exercise, we are just going to remove it for the time being. Okay, R273 will remain, and then oh, okay, and then you have all the different splice mutation will be excluded. Okay, and then wild type will be remain here. So that's it. So now we have a final data set that we can do some calculation with Fisher exact test and so forth. In this exercise, we are going to use the Microsoft Excel to conduct a Fisher exact test. Now, there are many ways that a Fisher exact test can be conducted, but when you want to apply it to a big data set like what we are doing here, I think the option for the Fisher exact test add-in would be one of the valuable one to try on. Okay, now let's come back to the Excel sheet that we have downloaded and also prepared just now. So what we are going to do now is we are going to prepare a few extra column, basically to count the number of mutations that are associated with different P53 status. So I'm going to create here, which is P53 wild type, and the target gene can also be wild type or mutated. Okay, similarly R273. So it could be like this. So I'm just going to create this uh, few more column here. So now imagine this is basically quite similar to a 2 by 2 contingency table that you would normally do in your Fisher exact test. So, but this is basically created in a row basis so that we can then use the Fisher exact test add-in to actually calculate it across all the data set. Imagine, okay, so they are all together close to about 40,000 genes here, and well, if you want to do this one by SPSS, that is almost impossible to do it. Okay, so now I'm going to prepare this column here. Now, the number of mutation is basically equals to well, to the sum of the whole data set in the P53 wild type tumors. So in this data set here, the 0, okay, basically represent no mutation in this particular genes, whereas if there's a 1 means that there are mutation in this particular genes. Now, always bear in mind is that the PanCan12 dataset or the PanCan12 mutation dataset only identify mutations that are missense. So in other words, is that only missense mutations are included in this data set. Silence mutations are not particularly picked up, and it will normally designated as 0 as well. So do bear in mind with that. Okay, what we are looking at here and analyzing here only limited to missense mutation. Okay, so I'm going to calculate how many cases of mutation in the wild type P53 for this particular gene. So what I'm going to do is I'm just going to sum up okay all the mutations in the wild type uh, tumors. So okay, the wild type start from here all the way to this end here. So this is that. So there are 4 mutations of this UBE2Q2 genes in the P53 wild type tumors. Okay, I'm equally going to calculate it for R273. So just going to select the cell, right? So these are already R273 mutation. Okay, and also for um, R175 as well. Okay, now I just discovered there is something here which we forgot to delete. I think there is a mutation class which was designated as others. See, well, we left that out just now. So I'm going to delete that. Okay, and then now the R248. Okay, I know that it is not included in this study, but what in this analysis I include that in is basically just to look at whether there's any things coming out from the R248 as well, because it is also one of the hotspot mutation, as we have indicated in the first exercise. So I'm going to include that in our analysis, and also the NS. Okay. Okay, so this is basically the number of mutations, okay, associated with each P53, what we call it, P53 status. Now, if I want to, the next thing what I want to do is basically I want to know the how many cases are the wild type uh, genes that are associated with different p53 mutations. So what I'm going to do is basically I need to know the total number of wild type cases minus of the cases that are mutated, then that will give you the number. Okay, here. Now, to know the total number of cases that are wild type or R273 or 175, there are many ways you can do it. One way is you can copy and then paste it into a separate sheet and then count it. Or you can simply use another good Excel formula. It's basically you can put equals to count if as, so they will tell you is that in this function, okay, it will count the number of cells specified by a given set of conditions or criteria. So it's good. So we are just going to use this formula here. Now it will then tell you, okay, you will need to select the range. So the range is basically from here all the way to the beginning. We can then uh, lock it, all right, and then the criteria would be wild type. Okay, this cell here. So that will straight away give you a total number, which is okay, 969 cases that are wild type P53. Then we can copy these cells, and then we can then it will then calculate how many cases are R273, and also how many cases are 175, R248, and also NS, and so forth. Now that we can use in order to know what is the number of genes that are okay wild type sorry, well, in order to know that what is the number of wild type UBE2Q2 that are associated with wild type P53, so we just need to use the total number minus of the number of mutated cases. Similarly, for R273 equals to 60 minus the mutated cases, and R175 equals to 47 minus the cases, and so forth. Okay. Okay. Once you finish calculating, okay, you will have a data set something like this. Next, I'm going to create a few more columns to calculate the p-value for the Fischer exact test by comparing whether the mutations in different class are significantly different. So I'm going to put that here, p-value, and this will be R273 versus, or basically R273 versus wild type, R175, R248, and also NS versus wild type. Now, to actually calculate the Fischer exact test, unfortunately, and in Excel, there's no existing formula that you could do it. One way that you can do it quite easily is that you can go to this website here, www.orbitfamily.com, to download an Excel add-in, okay, which can actually install into the Excel and then run the Fischer exact test in Excel. So there is a proper instruction for you as to how you can install this add-in into the Excel. So you can, well, please follow exactly, and then make sure that the add-in file is copied into the designated folder as it is. So in this exercise here, we will basically use this add-in which have already been pre-installed. So in case if you are not sure how to install it, okay, again, look for the instruction, and when you come to the Excel, okay, you can then manually add it in if you want to. So you just go to File and then Add In. All right. So here, see, Manage Add-in, Excel Add-in. You can just press Go, basically. Then you can browse where the file is. So this is okay. This add-in is basically installed in this folder path. So you need to, again, you need to make sure that it is installed in the designated folder path in order for it to work. So I will just select this. Okay, then it will tell you that it is already been installed, and replace it. Now you just press OK. So now, in order to calculate this, so now when you can put a formula now using see if Fischer exact test FET, then you just need to select the cells that you want to compare to. So in the first column here, I want to compare the mutations of a particular genes that are associated with R273. So I'm versus wild type. I'm just going to select okay the cells that is needed to do the mutation. See, this is basically, again, as I mentioned before, very similar to the contingency table that you normally seen in the common 2 by 2 Fischer exact test, right? So then straight away it will give you the p-value, and you will do the same for R273, but make sure it is always okay, adjust it and then compare it to the wild type. So you can select the cell like this, and then for R248, so okay, and for NS as well. So you can have this, this. Okay, so now here you got all the p-value. Okay, once you have completed all the calculation of the p-value for the Fischer exact test, you will have the result look something similar to this. So you will find some of the genes that are totally not significantly associated with R273 and so forth. So, but there will be some genes, for example, okay, we just do a quick filter check. Okay, some of the genes that are significantly associated, for example, we will set it as a p-value less than 0.01 to filter it in basically. Now, it will take a while. Yep. For example, here you will have, well, you will be able to find some of these genes. Basically, they are significantly associated with R273H, and you can then scroll it back here to look at it and ionically. So TP53, so should be TP53 is associated with R273 mutation, so that is very ionic, and also cross validate the analysis is correct. And interestingly, also have GATA 3 P10 and some of the and also beta catenin as well. So those are some of the well-known tumors associated genes. Now, one thing you always have to remember is that, well, the p-value of the Fischer exact test, it only tell you the association of a particular genes with a certain condition. In this case here will be the R273 mutation. It doesn't, however, tell you the direction, okay, where the association is, meaning that whether the particular mutation in the particular genes are more uh, common in the wild type, or it is more common to the R273 cases. In other words, what it means is that, well, the p-value doesn't give you a sense whether a particular mutation is actually co-occurred with R273 mutation, or if it is mutually exclusive in R273 mutation. So in order to do that, we need to create another few more columns to basically calculate that. And here, what I'm going to do is I'm going to calculate the mutation frequency of each gene in the different p53 uh, classes. So now, for example, R273 mutations. Oh, sorry. Basically, I also need to include the wild type as well, because we need to compare it with the wild type. Okay, let me disable the filter first. So the wild type frequency, okay, the mutation of this particular genes that are in p53 wild type tumors are basically equals to uh, the number of mutation over the total number, which is 969. Okay, so that is the frequency. So, and same for R273 is equals to this over 60, and R175 is equals to this over 47, and R24 is over 47, and NS is equals to this number here over 141. All right. So this is basically the frequency of the mutation of the particular genes. All right. Let me just uh, check in right decimal point. So then you can then copy this data and then paste it across. Oops. Okay, so now you have the frequency of the individual gene mutation. Now the next thing I'm going to calculate will be the log2, what you call it, log2 ratio. Okay, so and how am I going to calculate this? Basically by comparing it to wild type. So how many times differences in terms of the mutation frequency compared to the wild type. So to do that is basically here I'm just going to use log, okay, uh, so is equals to the mutation frequency of the particular gene in R273 over the mutation frequency in wild type P53, and so that will give you the ratio, and then you just log to transform them. Okay, so you will see some of the number it appear, what you call it, well it doesn't calculate because, well, this number is equally basically equals to 0 or infinity basically. So anyway, so we will continue to put in this formula again. So every time you put in, it will be compare versus the wild type. So all right, so that is how it is. Okay, and then you copy and paste the whole cell again. So now you have some numbers here. Now, so if you want to look at this data set more carefully now, so let me filter it. So you can first filter by looking at the p-value, make sure that it is by Fischer assay test, it should be less than or equal to 0.01. And these are the genes that are significantly associated with R273 mutation. Okay, here you go. These are all the genes that are significantly associated with R273 mutation. And if you want to know the direction of it, then you can start by looking into this value here. Basically, a positive value indicate that it is a co-occurrence with a R273 mutation, and if it is negative, then it indicates that it is basically mutually exclusive. Okay, so for example, what you can see here is that, well, let me go to the P53. So for example, P53, you have, well, you have a very good co-occurrence, obviously. Now, but whereas for if you look at P10, okay, here this is a P10, which have a negative number here, meaning that, well, it is actually mutually exclusive. What it means is basically when P53 is mutated, or when R273 mutation happens, most of the time, okay, the P10 will remain wild type or vice versa. That is what it means by mutually exclusive. Now, before we move on, you will notice that there are a lot of cells that have indication with the hash number or hash div. So how do this come by is, for example, okay, we will focus on the hash number first. Now, the hash number come by is basically during when we calculate the ratio. So, and this will happen basically, for example, in cases where you have zero mutation frequency in the R273 uh, cases versus those a positive value in the wild type. So when you calculate the ratio, it's basically equal to 0 over 0.0031, and that will give you 0. And log 0 would actually give you an error because you won't be able to calculate the log 0. So now, these are quite some of the extreme cases. So if you go back to your number of cases again, so for example, okay, here looking at CHMP12, CHMP 1B. So if you go back here on the number of cases, what you can see is basically you have 3 cases of mutation in the P53 wild type cases, and you have 0 mutation in the R273. What it means here is that, well, those cases that when you calculate the ratio have a hash number are actually mutually exclusive cases as the extreme value. So now to do that, I'm not going to, well, one way to conserve this information is that we will still give it a direction whether they are mutually exclusive or co-occurrence, but but the magnitude of it, well, is questionable in this case here. But nevertheless, what we are going to do is we are going to assign this hash number to a maximum negative value. So if you look at the range here of this whole data set, basically the range is from -3 to uh, to positive 5.01 basically. So I'm just going to introduce an arbitrary number of -6 in this type of cases. So I'm going to basically replace this into -6 just to indicate that these are all the cases that uh, that are actually what you call it mutually exclusive in the extreme value. Okay, so now we have replaced all the hash number cases with a -6, which indicate a mutually exclusive. Now, now we have to focus and deal with this hash div. Now, hash div is a little bit complicated. There are two conditions where hash div could occur. One of it is 0 divided by 0, which give you infinity. Or another cases is that, okay, is that you have a particular value divide by 0 and you will also get infinity. So to do that, what I'm going to do now is basically to look at those cases that are actually what you call it 0 over 0. So I'm going to basically filter this as 0. Okay, mutation frequency is equals to 0. And then the R273 is also 0. Oops. Okay. So, oops, sorry. I'm just going to select only those 0 cases and also the R273 which are 0 cases. Okay, so now you will see that okay, all these cases here that indicate the hash div are actually the uh, 0 over 0 cases. So there are no mutations whatsoever in the particular genes, either in wild type or in R273. So that is what it means. Basically, I'm just going to replace all this with an arbitrary number of 0 here. Okay, and then I'm going to do this for the rest of the set for R175, R248, and also NS as well. Okay, so this is how it will look like after you have replaced a hash div in the 0, 0 cases into the arbitrary value of 0. Now, the next thing is that while you will still get some value which is hash div here. So for example, okay, like I just select the hash div. So you will still have quite a number of hash div uh, cal calculation error. And now those that are remaining, however, are actually the one that are core occurrence with R273. Remember, okay, these are the cases is that occur because, well, you have a particular value, okay, over 0 that give you infinity. So these are the cases that are mainly core occurrence. So what I'm going to do is that the remaining hash div cases would be representing core occurrence in the extreme value. So what I'm going to do is I'm going to now use, okay, safely replace all this into a arbitrary number of positive 6, which is the maximum of the whole data set. All right, we will replace that. So it will take some time. Okay, finally, if once you have calculated everything, so it should look something like this. And to interpret this data, what you can do is you can filter again by p-value of less than 0.01. So this will be all the cases that have significant association with R273. And now here you have all the data that representing the direction of the association. For example, if you want to look at cases that are uh, what you call it uh, core occurrence. So basically, you will look at those cases that are positive, which is more than 0. Okay, so you can filter that out. So here are basically, and you scroll it back here. So then you will get all these genes that are basically core mutated with R273 mutation. And vice versa, if you want to look at mutually exclusive cases, then you will select less than 0, which is the one that gives you all the uh, minus or the negative value. So you have 4 genes there, so that include okay P10, beta-3, beta-catenin, and MAP3K1. That's it for now. Thank you. In this exercise, we are going to plot a minus log p value obtained from the Fischer exact test over the log ratio in a scatter plot. Okay, we come back to the well, we come back to the Excel sheet that we have prepared earlier by looking at the mutation frequency and also the Fischer exact test p-value. Now, the next thing what I'm going to do is I'm going to calculate the minus log p-value for all the data. So now, basically, to do that, what I need to do is I'm going to prepare a few column here. So this will be your minus log p-value. Okay, so you just basically put in the equation minus log 10 p-value. So this will be here, and then you just copy and paste all the way over. Okay, so all right, here you go. You can then arrange it accordingly. All right, so now what you have here is that, well, you have the minus log p-value and also the log 2 ratio. So to plot a graph, now what we can do is we can plot a scatter graph. So you just go to insert here. Okay, so then you go to scatter. So this is a scatter graph. So now what you need to do is basically we are going to plot 3 series of graph to show some of it that are co-occurrence and some of it that are mutually exclusive and some of it that are not significant changes. So I'm going to copy this graph over into another page here for easy access basically. Now, so here I'm going to just take the log ratio of R273. So put it here, and also the minus log p value for R273 mutation. So what I'm going to do now here is basically to plot a graph, okay, using all the data that we have. So I will all, and then x value would be the log 2 ratio, all right, and then the y value would be the um, minus log p. Okay, that's good. So this is how the graph would look like. Oops, we might need to change the graph type slightly different. So I want just the scattered basically. Okay. Now this is quite interesting here. There's one data point which has a very high log p value. I bet that must be the p53 basically. Okay, let me just check that. Yes, that is. So you will see that while the p53 is exactly high value there. So now, so what I'm going to do is I'm going to remove the p53 data point for okay for the moment because um, okay. Okay, that's good. Okay, now I'm going to input the same list again without the p53. Okay, so here's much better here basically. Um, oops, place value only. So this is much better basically. So now what you can see here is that, well, this is how the so on the y-axis it will be minus log p value, and also the at the bottom here that will be the log ratio. So again, how you want to interpret this is that, okay, if it is positive log ratio, then you will actually have a co-occurrence, and then if it is negative, then it is mutually exclusive. All right, so that's good. So now there's another thing which I want to plot would be this would be the one that are significance association. So I'm going to put select, basically filter by p-value. So this will be all the significant, statistically significant association. Okay, genes that are significantly associated with um, different p53 status. So, all right, and I'm going to choose those that are co-occurrence first. Okay, greater than 0. Now, so this is the list of the genes that are co-occurred. So I'm going to copy this to here, and also genes that are significantly mutual exclusive, less than 0. So which is just 4 here. Okay. So, and in the subsequent uh, okay, now I'm just going to add another series here. Basically, these are log2 ratio, and then this would be log minus p. So this is a significant co-occurrence gene. So I can name this as co-occurrence. And then I will have another list which is mutually exclusive. Oops, sorry. Mutually exclusive genes. So which I only have 4 here. Yeah, all on top of here. Okay, so now, so to make this look nicer, so I'm going to format this into uh, well, into a different color shade. So we can have so okay, we can have the line or the markers. So I'm going to change this into just a gray color. Okay, and then oh, basically I putting it to no fill, and then the line. So okay, the border would be gray color, but then the marker would be white color. So it will be something like this. Okay, and then I will have one group. So these are the co-occurrence group. I'm going to format it differently again. So no line. I'm going to format the marker into okay, no fill, but then borderline I'm going to put that as red color. Okay, and then another one. So this is the mutually exclusive. I'm going to put it into, let's say, green color. Oops. All right. Okay, no fill, and then I'm going to put border as green color. Okay, so that's how it will looks like. Basically, you can readjust the graph accordingly, and then put it into the PowerPoint slide as you like. Okay, so what you need to do now is basically to repeat the same process with all the different p53 mutation, plot this type of graph, and then put it into the PowerPoint. Once you finish plotted the scatter plot, so you should be able to get a diagram, something that will look something like this. So the green basically indicate those genes that are mutually exclusively associated with the R273H mutation, whereas the red are basically indicate those that are co-occurred together with the R273 mutations. Okay, to move on, the next exercise, what you can do is you can use the same data set and do the same analysis to plot what we call a circular genomic map using the mice circles. This is one of the way, and in fact, well, it's quite a good way to actually show, particularly in terms of the gene amplification and also deletion in the whole genomic map, to look at what is the region that are particularly mutated or particularly deleted or amplified as well. Okay, so to plot the circular genomic map, there are many words, there are many programs that you can use. One of the more popular one is something what we call Circos, but unfortunately Circos is more of the script or program based, so you need to key in the script one by one, and it might be quite complicated for beginners. So there's another alternative which I just found out recently, so which is a good starting point. It is called the My Circos, basically. Okay, if you just go Google My Course, so that it will come up. Now, the My Course, okay, you need to register. So registration is free. So now, if you go to My Course, there's a very simple interface, especially if you are just doing human genome. So in the following session, I'm going to show you from steps by step, okay, how you can actually input and prepare the files to be uploaded to MyCircos to generate a circular genomic map. So if you want to learn more about it, so the help set, you can always go into the help session here, which is very helpful, and then you can read up more about different kind of customization that you can make the graph look much more nice. Okay, so under this interface here, the first thing that you want to do is basically to prepare a file that the links file in the text delimited format. So this will be the links that will basically establish the association line between one gene to another. If you are not sure how to create these files, you can go to the help here. They will tell you, okay, all the information that's needed. For example, for links, so you need to have different columns here, basically from which chromosome it start, okay, start from what position of the chromosome and then to which chromosome and then which start region to the end region. So what I'm going to do is I'm going to show you how you can actually create this quite conveniently in the Excel file. So here I'm just going to copy these are the required data into the Excel sheet, basically. All right, so you can basically copy it that way, or you can copy into the what you call it. Okay, and then you can change it into different column. So tab delimited, that would be space. Okay, so now into each column. So now to do that, so the next thing what we want to do is we want to identify, okay, basically from which region from which gene uh, that we want to establish the link to which other genes that we want to look at. So in other words, basically when you look at the circular genomic map, so normally you will see the association line. So what we will do here, basically the from in our case will be mostly the P53, and to would be to the genes that where the association is actually located. And to do that, okay, we actually need to find out where is the location of the gene. So now, so if I may go back to this list here, basically. So what I'm going to do now is going to show you how you can actually plot the uh, genome, what you call it, the circular genomic map in the genes that are associated with R273 mutation. So again, so we are going to filter by p-value. So these are the genes that are significantly associated, so not a lot. All right. So and then we come back here. So we are going to first look at co-occurring genes. So must be greater than 0. All right. So these are all the co-occurring genes. So we are going to copy and then paste it here. And then we are going to look at the genes that are, what we call it, less than 0. So these are the mutually exclusive gene. All right. Sorry. Okay. So now we have two lists here, and of course we also have TP50, 53. Okay. So good. So now, what we will do now is basically we want to find the location of these individual genes. You can go to the what we call the genome browser, which is curated by the UCSC. You can go to the genome browser here. well, what we are trying to do now is basically to identify the location of each of the particular genes so that we can plot the graph. So you can go to then click on the table browser. Okay, once you go into that, so we make sure that all the data is correct. So we are going to look at human genome, the assembly. This is the latest assembly. All right, so now we will click that at genome. So okay, I think everything is good. So now what you can do here is basically the identifiers. So this is where you can actually copy and paste your gene list. Okay, so you can copy, let's say this is the associated genes and also the other one which is here. Then you can actually submit the table. So once it is submitted, so then you can get the output. It will come back as a plain text format. All right. So you can copy this into your notepad first. Okay, and then you copy this again, okay, into the Excel. So here you go. So basically, what it tells you here is, for example, okay, this is the gene name. So uh, where it is located at chromosome 1. Okay, start from this position and end at this position. Okay, so you also see that there are actually sometimes more than one entry of this particular gene because of the different isoforms. So in this case here, we are not so we are not so concerned about the so-called isoforms. So what we are going to do now is basically we are going to remove some of the redundant names, okay, gene names. So what we are doing now is basically to sort it, okay. So we don't need this information here. We can delete. Then after that, we can use a formula to identify which one is actually redundant. So if, so you can use the if function. If this cell is equals to the previous cell, then it will be 1. If not, then it will be 0. Okay, so you copy all this down. Okay, so then now you have a bunch of 0 and 1. So basically 1 indicate that, okay, this cells is equals to the cells preceding it, and also this is equals to this. So those basically tell you these are the redundant gene list, gene names that you can actually delete it. So what I'm going to do is, okay, I'm going to select all the ones, and then I'm going to delete all this. All right. Okay, and here you go. And finally, okay, you would have all the gene lists, okay, which is with single, what you call it, a single entry only. So what I'm going to do now is basically, okay, we know here TP53, okay, so now, okay, so what I'm going to do now is basically copying, we don't need this information, so we only need the chromosome number, where it is start, where it is end, and the rest of it we actually can delete it. All right, so I'm going to copy this list and then put into the Circos. So let me put it into the Circos list here. So it will be, oh, let me just move it. So I'm just going to copy this list and I'm going to put it here. Oops, sorry. So I'm going to put it here. Okay, so the gene names is basically not required. So, but we can put in here another one option. I will come to that later on. So basically, okay, all the association would be from P53. So which is this one here. Okay, and then to the list of genes that are associated with the what you call it, the co-occurrence. So to do that, basically, okay, P53 already took it out, so I'm going to delete it from here. Now, the next thing what I want to do is basically, okay, I'm going to change the thickness of the line. So anyway, so well, I will come to the option later on. So we will just plot the circos map first. All right, so I'm just going to copy this all the way down. Okay, and then you can copy this and then put it into, copy and paste it into the text file. So you can save this. So, okay, let's say if I save this in this circos text, all right. So then over here, if I come back to the circos data here, then I can actually load this file. All right. So circos text file here. I'm going to designate this description. So I don't need to display any histogram. If you want to add any more tracks, you can, but in our case, well, uh, we are going to just do a very simple circular map, so we don't have to display many track here. So then you press the button generate. Okay, once it is finished, so you will get a circular map, something like this. Okay. Right, so with the chromosome number being labeled here, and this is where P53 is, and each of this line indicate there's an association between P53 and the particular target genes in the different location of the whole genome. Now, this is a very basic circular map, so it might not be so informative. So the next thing and the next thing you can do is basically you can actually customize it by coloring the different association, whether they are co-occurrence or mutually exclusive. And you can also increase the thickness of the line here to make it much more prominent. And to do that, basically, we will go back to the Excel file here. So you can create one column here, which is called option. So now, okay, if you want to learn more about the option here, you can actually go back to the home here, and then you can. Oops, sorry. You can. Sorry, you have to sign in again. Okay, you can go back here, and then it will tell you, okay, how you can actually. If you go to the tutorial here, it will tell you how you can actually change the customization. Okay, next what I'm going to do is I'm going to put the option here to show to customize the display of the link and of the color and also the thickness. Now before I do that, I just want to identify, okay, under this list here, so these are the 4 genes that are actually mutually exclusive. The others are basically what you call it, co-occurred with the R273 mutation. So now what I'm going to do now is basically you just put in a command here, color equals to red or any color you want. And the thickness, okay, you can change it whether you want, depends on how thick you want. You want 5 points, 10 points, it's totally up to you. You can put it there, okay. So I'm going to copy all this, okay, to all the, what you call it, the co-occurrence mutation. Okay, and then for the mutually exclusive genes mutation, I'm going to color it as, let's say, blue color, or if you want, you can use green color to accommodate with your scatter graph. But basically, I would prefer to use the blue here just to show you the example. Okay, so that's how it is. Right, then after that, you will copy this again, and then you put it into the text file. Make sure you save it, and then you can re-upload your text file again. So then you press the generate button. So it will take a while. Okay. Okay, here you go. This is a new circular map with the customization that we put in just now. So the red color line here indicate those genes that are basically co-occurred with R273 mutation, whereas the blue line indicate the genes that are basically their mutation is mutually exclusive. Okay, when compared to R273 mutations. So now the next thing we want to do is that we want to download this file and then okay, so the graphic file and then to put into PowerPoint. Now, unfortunately, if you notice that, okay, the file format is actually what we call SVG. It's actually a vector file format, which unfortunately PowerPoint do not recognize it. So what we need to do now is basically we need to convert this SVG file into something that PowerPoint can recognize. So there are many software that you can actually use. You can Google it, and one of it is called the SVG PNG converter. So if you go to here, okay, so it's just fvg to png.com. So if you press the upload button, so you can choose to upload the SVG file. Okay, yeah, where is the SVG file? Okay, let me see where it is. So, okay, 93287. Okay, again. Okay, here you go. So this is the file that I've downloaded previously. Okay. Now that it will convert it into a PNG file, which you can actually insert it into PowerPoint quite nicely. Okay, then you can download this PNG file. Okay, you see, so it's downloaded now. So okay, this is how later on, if you want, you can just insert picture uh, from the PNG file that we just convert we just converted basically. All right, here you go. See, now you can insert the whole file over here, then you can actually resize as you like. Okay, so that's it for the plotting. In the last part, then we are going to look at is basically how to analyze this based on the pathway analysis. Okay, so by the time when you finish plotting all the circular genomic map, so you will get something similar to this. Also remember to put in the legend to show that the red color line indicate co-occurrence, whereas the blue line indicate mutual exclusivity. Okay, now finally, in the last part of this exercise, we are going to perform a pathway analysis of the genes that we have identified to be significantly associated with different p53 mutation. Okay, so to do the pathway analysis, we will come back to the Excel sheet that we have. So this is the data that we use to plot the circular genomic map. So what we want to do now is basically to look at the genes that are associated with R273 mutation. So this is the list of the genes that we have identified. Now, what I'm going to do is basically I'm going to copy this gene list. Now, note that while you don't have to be bother too much about whether they are co-occurrence or mutually exclusive, so because in a pathway, each of the genes will have a different role, whether they are activator or inactivator. So for in this exercise, basically, I just want to know which pathway is particularly dominant that might be regulated by the R273 mutation. All right, so I have copied this list here. The next thing what I'm going to do is I'm going to go into this web page, okay, called the Consensus Path DB Human database. So or the CPDB. Now, there are many ways that you can do pathway analysis, basically. Now, one common one would be using David. But the David, one of the limitation is that you can only do one pathway the pathway analysis on one single database, one at a time. Whereas consensus path DB, you can do it all together. So what I'm going to show, okay, this is how it works. Basically, you go into over representation analysis. Okay, once you click on that, then you paste your gene list here. So make sure that you identify the gene identifier, which is basically gene symbol, and then you press the submit button. Okay, then it will land you into second page here. There are many options that you can choose. For this exercise, we will just look at pathway only, and by default, the p-value cutoff is actually 0.01. What it means is that basically any pathway that are more than 0.01 would not come out in the results later on. Okay, so here I'm going to press this button and here you go. So you get a list of pathways that are associated with the gene list that you put in. For example, endometrial cancer, you get the p-value here, the q-value, and it's also tell you where the pathway data source is. In this case here, this is based on the CAG pathway database. You can also have some of it which is from PID, Reactome, and so forth. Now you can read up more about the pathway source or the pathway database. One thing about the pathway source is that every single database would have some different or some unique gene set that is associated with a particular pathway. So you got to be very careful to look at that, because sometimes, okay, even though, for example, the same pathway that might be differently represented in CAG or in PID, so you need to be very careful about it. So anyway, so now we got the pathway that are basically associated with the gene list that we put in. So I'm going to select all, and then I'm going to download this. All right, then it will come into a text file. So as usual, I'm going to copy this, and then I'm going to put this into the Excel. All right, so now that you have, now you have the list of the pathway and also the genes that are the genes that are overlap between your gene list and also the one that are curated in the pathway, as well as also the p-value and q-value as well. Okay, so just to cut the story short, basically here I basically uh, conducted all the pathway analysis for all the mutation that we have, and here I also calculated the minus log p value for us to plot the graph later on, basically. Now there are many graphs that you can plot if you want to show this data set. One easy way to plot it would be, for example, just using a column graph. Okay, you can put it here, and then you just select the data, which is basically the series name, or you can just here you can select the series name here and also the p-value. Okay, so now you can draw a chart, a bar chart, something like this. Now, but the major problem with a bar chart is that on the x-axis, so your label most of the time, if it is too long, then it will be truncated. So now it's up to you, well, whether you want to represent it in a bar chart. So if not, then you can represent it in other way. So one of the way that you can represent, but not quite commonly used in the scientific literature, is something what we call the tree map. So in the tree map, for example, okay, it can give you the data set that you have in a way that, well, the size of each of these square represent the proportion of the value. So now, if you want to look at it carefully, so now there's one way that you can set it. For example, okay, you see that. Okay, the different size of the square basically represent the different value. In this case here, the log p value or the minus log p value here. So if you want, you can even customize it. For example, by adding the data label, you can also include in, for example, if you click on label option, so you can actually label the value here. So all right, if you label in new line, so you can see that now. So this is the first pathway that is associated with R273 is endometrial cancer pathway with the minus log p value of 4.48 and so forth. Now, if you change the value, okay, instead of log 10, you can use log 2 so that the differences will be much more bigger. Oops. Okay, I'm waiting it to come back. Okay, for example, if you change it to log p, then the value will be slightly, I mean, the differences between the value will be slightly bigger. So you will have a clearer sense to look at it. But anyway, so either log 2 or log 10, I think that's fine. So this is just one way that you can represent the data set. Okay, and here this is just an example of the graph that I have plotted for the R273. Okay, this will be R175 and also NS. So what I did is basically I represented it in the tree map so that the audience or the reader can actually look at each of the pathway here more clearly. More clearly. At the same time, I also summarize it basically in a table format so that they can also see the individual genes that are in our gene list that are associated with that particular pathway. So what I did also is that I labeled the pathway by number so that on the table here that you can just use the number to refer to it rather than having a long pathway name. So that's it for the pathway analysis. Okay, finally, well, this is just to summarize basically. Okay, what I've shown you so far is basically just to look at the analysis of the mutation data set by using the Fischer exact test, and also show you how you can use a scatter plot to actually show the log p value and also the log ratio to identify the genes that are associated with certain p53 mutation, and also use the circular genomic map to actually indicate whether the association is co-occurred or whether it is mutually exclusive. Now, in the subsequent exercise, okay, what you can do is you can apply the same principle to analyze the copy number variation, the DNA methylation, mRNA expression, and also protein. Now, for copy number variation, okay, one way you can analyze it is basically you can apply the data by using a copy number cutoff. If you look at the nature of the data set, it is actually in a continuous data basically. So all you can do is basically you can use a copy number more than one or less than one as a cutoff to indicate whether that particular case have amplification or deletion. In general, basically in the TCGA, so if it is 0, that means there's no copy number variation. If it is 1, that means it is probably one of the alleles being amplified. And if it is 2, that means that it could be homozygous amplification, or more than 2, basically you have amplification of more copies of the particular genes. So what you can do is you can convert the continuous data into discrete data of 1 or 0 by applying the copy number more than 1 or less than -1 as a cutoff. Then you can actually perform the exact test quite easily. Now, be careful is that basically, well, when you look at co-occurrence and also mutually exclusive in the copy number variation, you need to consider, okay, whether you are looking at amplification or deletion. So these are some of the hints that, well, you need to pay attention to. So I will let you to try around with the data set to look at that. Now for DNA methylation, basically the data is already come in as a discrete, meaning that it's 1 or 0. So basically you can apply almost a similar concept as how you do with mutation. Now, but for mRNA and also for protein, so it is slightly different. So because mRNA and the protein, basically the data is actually continuous, so it is not discrete. What you can do with those type of data is that you can use a student t-test. Now, they are also good to try out whether okay, you might want to do a log2 transformation if the data is not normally distributed before you calculate the student t-test. So I will let you to explore a little bit about it. Now, when you plot the scatter graph, okay, so I will highly recommend you, again, you will use the minus log p value as a y-axis, but the x-axis, okay, you can just plot it with the z-score differences, because the z-score is not linear in a way, so it will be very difficult to actually use the ratio concept to actually calculate the z-score ratio. So, and what you will end up, you will end up with a group of genes that are upregulated or downregulated. So that is pretty much it, basically. So so once you completed this part 2, then we will continue on with the part 3, which will be looking at how we can actually integrate all this data together.